Good afternoon, everybody. Thank you so much for joining us. This is Emily piping in, the chair of this year's Hanover committee. I'm going to have Dave McFarland introduce Georgios, but I just wanted to remind everybody to turn off your videos because this is going to be recorded. But I think it's probably okay to turn them back on during the question and answer period, which I will let Georgios and or Dave facilitate when we get there. So thanks again for joining us, and I'll turn it over to Dave.
Okay, thank you, Emily. All right, so welcome everybody. I know people are still popping in here. And so it's my pleasure to introduce Mr. Georgios Arseniou, who is a PhD candidate in the forestry department. And just to give you a little background on Georgios, he has a Bachelor of Science in Forestry in the Natural Environment from Aristotle University of Thessaloniki, Greece. So a student of Aristotle, very, very prestigious. He has a Master of Science in Photogrammetry and Geoinformatics from the University of Applied Sciences of Stuttgart, Germany. And now he's working on a PhD here, as I mentioned. And we've been doing very interesting work in the lab, trying to understand the form and function of trees. And Georgios has been collecting some very interesting data, novel data really, with a terrestrial laser scanner on urban trees. And he's going to share with you some of what he's learned.
All right. So thank you very much, Dr. McFarland. And it's my great pleasure to be here today with you and present you how we can use terrestrial laser scanning and digital image analysis to quantify the above ground surface area of urban trees. This presentation is based on a study that Dr. David McFarland from the Forestry Department at Michigan State University and Dr. Dominique Seidel from the Department of Silviculture and Forest Ecology of the Temperate Zones at the University of Göttingen in Germany and me recently conducted and submitted for publication. So, okay. All right. So to begin with, I would like to emphasize that the urban forests constitute a critical component of the global forest network. And we know that urban trees provide a wide range of significant ecological and socioeconomic benefits to human societies. So, for example, we know that urban trees provide shading, water purification, air pollutant uptake. They support the wildlife habitats in cities, and they're also important energy benefits for buildings. So in order to optimize these benefits, we need a better understanding of the physiological ecology of the urban trees. And the total surface area of trees actually is a critical component of the ecological physiology of the urban trees. The total surface area of trees consists from two main parts, the photosynthesizing part and the non-photosynthesizing part. So the photosynthesizing part is actually the leaf area that relates to the physiological processes of photosynthesis and respiration. And the woody surface area of the trees relates to the respiration of the non-photosynthetic tissues of the trees, because respiration relates to the cambial sheath and the living annual growing rings around the dead headwood of the trees. So both the leaf area and the wood surface area really influence the carbon balance of trees and their sustainable growth. Now, there has been a lot of work in how can quantify the leaf area of trees. And there are many methods. Digital image analysis is one of these methods that we have actually implemented in our study here. We can also estimate leaf area from hemispherical photographs or using gap fraction analysis by considering the porosity of the tree crowns. We can also use tools like the LAI plant canopy analyzer to measure leaf area, or we can do destructive sampling, which means that we cut the trees down and we directly measure the leaf area of the trees. And of course, we can build allometric equations to estimate leaf area from allometric variables of trees such as dBH or the total height of trees. And of course, there is the laser scanning technology that gives us the opportunity to create a three-dimensional representation of a tree. And using terrestrial or portable laser scanners, we can estimate the leaf area of trees. So the empirical leaf measurement has become a major focus of scientific inquiry. However, the quantification of the woody surface area of trees is more challenging. Of course, there have been other studies in the past that focus on how we can estimate the woody surface area of trees using destructive sampling, where people were cutting trees down and they dissect them into pieces, and they measured lengths, diameters, and thicknesses. And from these measurements, they were building allometric equations and models to estimate the woody surface area of trees. In other studies, people have used geometric approximations of stem and crown shapes using, for example, conic or parabolic approximations. We also know that branch surface area estimates can come from vertical photographs. And also in previous studies, people have used the principles of pipe model theory and the branch divarication theory to estimate the woody surface area of trees. However, in all these methods, there is a challenge that all these methods face, which is the difficulty to measure the total woody surface area of trees due to their structural complexity or their fractal-like pattern. And we know that trees can be treated as fractals. Actually, Leonardo da Vinci was the first who observed that the cross-sectional area of the branches is preserved across the different branching orders, which actually explains the crown self-similarity of trees and the structural complexity of the crowns of the trees. So the crown self-similarity means that through the branching network of a tree, every branching point looks the same whether we look at the first or the last branching point. And according to metabolic scaling theory, there is a set of rules that describe the architecture of these ideally symmetrical and fractal trees, where, for example, we assume that each parent branch has two daughter branches, and each daughter branch has half the diameter of its parent branch and 1/3 of the length of its parent branch. However, trees in nature cannot fully express their inherent fractal character due to the environmental conditions in their local growing environment. And competition for light is one of the main factors that disrupt the inherent fractal character of trees. Therefore, it has been suggested to better treat trees as disrupted fractals. And we assume that urban trees have a more evident self-similar character compared to the rural forest trees because they grow in open and they either face very limited competition from their neighboring trees, or probably they face no competition if they grow totally in the open. And this was another reason that motivated us to study trees that grow in cities. Now, terrestrial laser scanning is a technology that gives us the opportunity to study the architecture of trees and their fractal structure. Terrestrial laser scanning is an active remote sensing technique where the laser is mounted on a tripod and it emits tens to hundreds of thousands laser pulses per second. And when these laser pulses hit their targets, which can be, in our case, woody material or leaf material, then this return energy is recorded by the laser scanner, either as a function of time or as a function of shift in the phase of the light wave of the emitted laser beam. And at the end, we have a detailed representation of the three-dimensional structure of a tree, which is a point cloud. So here you can see a picture of a point cloud of a tree in the figure on your right, which is a Cladicea triacanthus tree that I scanned in summer 2019 on campus, and it's part of our study. So you see that in this point cloud, the laser data captured the branching architecture of the tree, but also the leaf area and the foliage. So now that we have a basic theoretical background, let's focus on the research questions. Our first question is, how does the tree surface area distribution between leaves, stems, and branches, and the allometric ratios between component surface areas and volume help to understand the anatomy of trees in light of major theories of tree structure, such as the pipe model theory and the WBE theory, or the West, Brown, and Enquist theory. Second question is, how does the ratio of woody surface area to one-sided leaf area of trees, which is a measure of production efficiency, relate to the ratio of woody surface area to the total woody volume, which is a proxy for respiration rate? And finally, how does the external surface area of the trees relate to their fractal complexity and to different Euclidean measures of tree architecture? So here in this study, when we refer to Euclidean measures of tree architecture, we consider metrics that describe the length of all possible paths from the base of a tree to each branch tip. In order to answer these questions, we sampled 57 trees of 3 different species of different functional types on a Michigan State University campus. So we sampled 18 Cladicea triacanthus trees, which are compound-leaved deciduous angiosperms, 18 Quercus macrocarpa trees, which are entire-leaved deciduous angiosperms, and 24 Metasequoia glyphrosoboides trees, which are needle-leaved deciduous gymnosperms. In the table here, I have summarized some key statistics of the size of the trees in terms of diameter at breast height and total tree height across all species. So you see that we have a wide range of sizes, both in diameter and height, for the 3 species. So in July and August 2019, I laser scanned and captured digital images, color digital images of the Cladicea triacanthus and Quercus macrocarpa trees during the leaf-on period. Then I revisited the same trees in January and February 2020, and I laser scanned the same trees during the leaf-off period. We also made sure that there were no pruning effects between July 2019 and February 2020 that could create a bias in our results. Furthermore, in March 2020, I laser scanned the Metasequoia glyphrosoboides trees during the leaf-off period. So for the woody surface area analysis, we have 57 trees of the 3 species, Cladicea triacanthus, Quercus macrocarpa, and Metasequoia glyphrosoboides. And for the leaf area and total surface area analysis, where the total surface area includes both the woody surface area and the leaf area, we have 33 trees from the 2 species, Cladicea triacanthus and Quercus macrocarpa. Now, I would like to explain you how we scan the trees. In order to scan trees, we use the Faro-Focus 3D laser scanner, which emits laser light of 1,550 nanometers wavelength, with typical beam divergence 0.19 millirads and range between 0.6 and 330 meters. These technical characteristics of the scanner enables us to capture good quality point clouds. In order to scan an individual tree, we always scan it from at least 4 different directions and distances in order to minimize the occlusion effects in the crown, because, for example, some branches can be overshadowed by other branches or leaves in the crown. And then we also use these registration spheres, as you can see in the image below, these white spheres. These are registration spheres that are common points between the different scans for each tree that can be used to spatially co-register all the scans for each tree and create one single point cloud for each tree. After we create this single point cloud for each tree, the next step is to filter the noise. And the final step is to segment the trees, which means to extract the tree from its scanned background. Now, the next step in our analysis was to create the quantitative structure models from the leaf of point clouds, or the QSMs. So the QSMs describe the three-dimensional architecture of trees by fitting cylinders to a tree's laser point cloud. In a quantitative structure model, the branch and stem topology are preserved, and the algorithm also separates between the stem and branches of different branching orders, and it gives information about the size, the location, the hierarchy, and the orientation of the branching network. So in this figure here, which is from the literature, you can see an example of a quantitative structure model where these cylinders have been fitted to the point cloud of a tree. And you can see that the structure of the stems and branches is preserved. In our study, I used the tree QSM algorithm to create the quantitative structure models of the leaf of point clouds. And this algorithm generates multiple QSMs for each tree. Then it selects the optimal QSM and computes a distribution for its allometric variable, such as woody volume or woody surface area. In this figure, I want to show you how we go from a leaf of point cloud to a quantitative structure model. So this is a tree from our study. In figure A, you see the leaf of point cloud of a Quercus macrocarpa tree. Then in figure B is the quantitative structure model of the same tree. And in figure C, we have a close-up picture of the generated QSM, where you see that all these cylinders have been fitted to the point cloud data. So the different colors here denote the different branching orders. The main stem is colored with blue. The first order branches are colored green. The second order branches are colored red. And of course, they're higher branching orders. So after we created the quantitative structure models for each tree from their leaf of point clouds, we also estimated the total woody surface area, which is the total surface area of the cylinders that were fitted to the leaf of point cloud of each tree, as you see in the figure here. And here, when we talk about the total woody surface area of trees, we really mean the surface area outside of the bark tissues. Next, we separated the surface area of the main stem from the surface area of the branches. And we further analyzed the distribution of branch woody surface area per branch order, branch-based diameter classes of 1 centimeter size, and branch-based height classes of 1 meter size. And finally, we also computed the total woody volume and the component woody volumes of the main stem and the branches based on the volumes of the cylinders that were fitted to the leaf of point clouds. And now let's focus on the estimation of the one-sided leaf surface area. So we captured color digital images of the Glidicea triacanthus and Quercus macrocarpa trees during the leaf-on period in July and August 2019. I used the Canon EOS Rebel T6i digital camera, and I captured minimum 2 and maximum 4 color images from each tree from perpendicular directions, as you see in the figure. The next step was to take these images and back to the lab and remove the background. So after we moved the background and we had in each image the crown of the tree, only the crown of the tree, we're able to estimate the one-side leaf surface area based on a method pioneered by Pepper and McPherson in 2003. Here I'm showing you a simplified form of the model that estimates the leaf surface area. which is a function of two variables, the silhouette area of the tree crown, which is the crown area divided by the image frame area, and the framed area of the tree crown in the photograph scaled to the actual size. Of course, there are more steps and more other variables that go into the leaf surface area, but this is a simplified form of the model. And at the end, finally, the total sided leaf area of the tree is the average one-sided leaf area estimate from all images of the same tree. We also computed some additional tree structural metrics. So from the QSMs, the quantitative structure models, we computed the length of all paths from the base of a tree to its branch tip, as you see in this figure here on your right, because we know the length of all the cylinders whose topological hierarchy is maintained in a quantitative structure model. So after we computed all these different path lengths, we calculated the path fraction metric, which is the ratio of the mean length of all these paths divided by the maximum length of these paths, which, according to Smith et al. In 2014, describes the extent to which a tree diverges from the ideal fractal character. This metric, the path fraction, takes values between 0 and 1, and as you see in the figure on your left, the higher the value of the path fraction, the higher the crown complexity of the tree, and the more fractal evident character it has. From this data, we also calculated the distributions of all these path lengths. So we compute the 25th, 50th, and 75th percentiles of the path lengths, as well as the minimum, maximum, and mean path lengths. And the next metric that we computed was the box dimension metric, which is computed directly from a point cloud of a tree, and it is based on the box counting method. So this metric describes the above ground structural complexity of trees based on the above ground plant material distribution of the trees. And it quantifies the fractal dimension of a tree. In order to compute the box dimension of a tree, we need to divide the three-dimensional space of a tree into boxes whose edge is iteratively decreasing. And we need to count how many boxes we require in order to capture the whole structure of a tree. So the box dimension is actually the slope of the regression line between the logarithm of the number of boxes that contain the points of a three-point cloud and the logarithm of the inverse of the box size divided by the edge of the length of the initial box that, as you see in the figure, it captures the whole tree structure. The box dimension metric takes values between 1 and 3, and the higher the value of the box dimension, the higher the fractal, the higher the crown complexity of the tree and its fractal dimension. Finally, we fitted some models for the surface areas, considering the structural metrics that I described. So in these models, AW is the woody surface area of the trees in square meters from the quantitative structure models. AL is the one-side leaf area in square meters from the digital image analysis. AT is the sum of the woody surface area and leaf surface area of the trees in square meters. Then B is the normalization constant in the three models. DB is the box dimension metric, which is unitless, and it describes the fractal dimension of a tree. L is one of the path length metrics in meters, which is actually a Euclidean metric because it's simply the length of all these different paths through the branching network of a tree. Then C and D are the exponent parameters of the box dimension, the length metrics. And S is the species random effect because we also wanted to consider how the different species affect the three surface areas. And now let's move to the results. So we found that the average one-side leaf surface area was about 26% of the total tree surface area, and the rest of it was woody surface area across all species. We found similar values for the two species that we had leaf surface area data. So 25% of the total surface area of Gleditsa triacanthus trees was leaf area, and 27% of the total surface area of Quercus macrocarpa trees was a leaf area. Of course, there was a high variation from individual to individual tree across the size range of the trees. Now, this finding here agrees with what has been suggested in the past, that the trees require a large area of respiring woody tissues to support their photosynthesizing leaf surface. Furthermore, we separated the branch surface area from the stem surface area of the trees. So we found that the branches comprise the greatest portion of woody surface area of the urban trees. And the ratio of the branch surface area to the main stem woody surface area ratio ranged between 4.3 and 38.6 with a mean value of 16.4 across all species. This result indicates a resource allocation decision of the different species that invest more resources into building the surface area of their branching network versus their main stem. And it agrees with what we know about the form of the urban trees, which is a squat form. That means that urban trees have a short trunk and expanded crowns in order to better adapt themselves in the urban environment because they need mechanical stability against wind loads in cities. And these results also agree with previous findings from other studies where branchiness of Scots pine, where branchiness is the cross-sectional branch area over the stem surface area of trees, it was found that it increased as the stand density decreased. We also found that the branch to main stem surface area ratio increased with the box dimension metric, which means that as the crown complexity and the fractal dimension of the tree increases, the trees invest more resources to build the surface area of their branch network compared to the main stem. We also found that the branch to stem surface area ratio differed significantly among the three species, with the Gleditsa triacanthus trees having the highest value of the ratio. Therefore, since this ratio can differentiates between the different species, we also suggest that it could be an important component of the plant structural economic spectrum that explains the structural variation of different species across a range for optimizing light capture, carbon balance, and mechanical stability. So here now we have the branch woody surface area distribution per branch order. We found that the branch woody surface area significantly differed among the different branch orders across all species and within each species. And as you see here, the distribution of the branch woody surface area across the branch orders followed a positively skewed distribution, with most of the branch surface area accumulated in the lower branching orders. So for the Gleditsa triacanthus trees, most of the branch surface area was accumulated in the third and fourth branching orders. For the Quercus macrocarpa trees, it was mainly accumulated in the second and third branching orders. And for the Metasequoia glyphoseboides trees, the woody surface area of the branch was mainly accumulated in the first three branching orders. Now here you see how the branch woody surface area distributed per branch base diameter. We also found that the branch woody surface area differed statistically among the different branch base diameter classes across all species and within each species. So as you see, the distribution of the branch woody surface area per branch base diameter followed a positively skewed distribution that was somewhat bimodal, because most of the branch woody surface area came mainly from the medium-sized branches between 5 and 10 centimeters, and the larger branches, approximately of branch diameter greater than 35 centimeters across all species. Now, these results mean that the largest portion of the total branch woody surface area was accumulated in medium and large-sized branches of lower branching orders. And we need to keep in mind that variation in branch woody surface area is related to the diameter of a branch and its position in the crown. These results can be interpreted within the framework of the bimodal theory and the WBE model. And both theories, despite the differences of their inherent assumptions, they both assume that trees have a fractal-like architecture, and that and their vascular system consists of several tubes that taper from bottom to top. And according to the WB model, there is a power function that connects the surface area to the volume of each tube. So the surface area scales with the volume of its tube, which means that as the volume of a tube increases, its surface area also increases. And this explains why the medium and large-sized branches of lower branching orders had accumulated most of the woody surface area, because typically these branches accumulate most of the woody volume as well. Now, in this graph, we have the vertical distribution of branch woody surface area. In the y-axis, we have the relative branch base height, which is the height of a branch in the crown divided by the total tree height. And on the x-axis, we have the relative branch woody surface area, which is the surface area of a branch, of individual branch, divided by the total branch surface area of a tree. So across all species, you see that the vertical distribution of the branch woody surface area followed a parabolic distribution. And for the Quercus macrocarpa trees, we see that the relative branch woody surface area was peaking around the mid of the crown of the trees. Whereas for the Cleditsa triacanthus trees, the relative branch woody surface area was peaking above the midpoint of the crown length. And for the Metasequoia glyprosoboides trees, the relative branch woody surface area was peaking below the midpoint of the crown length. Now, in order to interpret these results, we need to keep in mind that the distribution of branches and foliage is heavily influenced by the shading and sheltering from neighboring trees, so that trees can maintain a positive carbon balance in their leaves. Nimenets and Valaders in 2006 created numerical shade tolerance indices for several species, ranging from 1 to 5, where 1 means that indicates a species that is very shade intolerant, and 5 indicates a species that is very shade tolerant. So for our species in this study, the Cleditsa triacanthus trees was the most shade intolerant species, and the Metasequoia glyprosoboides trees was the most shade tolerant species. Now, this is important because it means that the branch surface area vertical distribution was inversely corresponding to the shade tolerance of the species. Because of the patterns of branch area peaking in the upper mid, middle, and lower mid crowns for the Cleditsa triacanthus, Quercus macrocarpa, and Metasequoia glyprosoboides trees. So we can assume that the self-shading of leaves and branches could be an important element of the branching architecture of the open-grown trees, along with their inherent shade tolerance. But there might be also some other factors that are in play here. Now, here we have the relationship between the woody surface area over the leaf area of the trees and the woody surface area over the woody volume of the trees. These are very important metrics because the ratio of the woody surface area over the leaf area of the trees has been suggested in the literature as a metric of production efficiency. And the ratio of the woody surface area over the woody volume of the trees is a proxy for respiration rate. If we follow the logic that, based on previous studies, that the leaf area over the leaf volume is relating to the photosynthetic rates of a tree. So the strong and positive relationship between the two ratios indicates that as the respiration rates of trees increase, their production efficiency should also increase so that they can maintain their sustainable growth. Otherwise, they lose vigor. Now here we have, I have summarized the modeling results for each surface area. So we have, I have indicated the models for each surface area with the highest explanatory power. So for the model that predicted woody surface area as a function of box dimension and the mean length of all paths from the base of a tree to every branch tip, with the addition of the species, had the highest explanatory power, but it was not the most parsimonious. The leaf surface area explained by the box dimension metric and the mean length of all paths was found to be to have the highest explanatory power, and was also the most parsimonious. And then the model that predicts total surface area as a function of the box dimension and the mean length of all paths, with the addition of the species, was found to explain the most variation of the total surface area. So it had the highest explanatory power, but it was not the most parsimonious either. Now here, I want to show you the simple correlations between the woody surface area and all the structural metrics that we computed from the point clouds. We found that the woody surface area of the trees was positively correlated with the box dimension metric, which actually means that as the crown complexity of a tree increases and its fractal dimension increases, the woody surface area of the trees should also increase. Among the other structural metrics, we found that the mean path length was the most strongly correlated with the woody surface area of the trees. The mean path length characterizes actually the size of the tree. So this means here that as the size of the tree increases, its woody surface area should also increase. And we have this nice distinction here between the box dimension, which is a measure characterizing the fractal complexity of the tree, and the path length metrics, which are Euclidean metrics. Now here you see the correlations between the leaf surface area of the trees and the structural metrics of the trees. So we found that the strongest correlation was between the leaf surface area and the 25th percentile of the path length. The 25th percentile of the path length is actually the frequency of the shortest path length from the base of a tree to its branch tip, which actually indicates that as the crown length of the tree increases, its leaf surface area also increases. And for the total surface area, we found that the total surface area increased with the box dimension, which means that as the crown complexity of a tree increases, which means that its fractal dimension increases, the total surface area of the tree also increases. And we also found a positive correlation, the strongest positive correlation between the total surface area of the trees and the 25th percentile of the path lengths, where again, the 25th percentile of the path lengths is the frequency of the short path lengths. And we see that as the crown length increases, the total surface area of the trees also increases. Now, we found that the path fraction metric, which is the ratio of the mean length of all the paths from the base of a tree to its branch tip, divided by the maximum length of these paths was not significantly related to any of the examined surface areas. However, the individual metrics of path lengths, which again, they're Euclidean metrics, they were all strongly correlated with the surface areas. We found that the surface area of the urban trees was increasing with the fractal-like architecture of the trees, quantified by the box dimension metric. And according to the WBE model, this means that this indicates that there is efficient respiration rate and sufficient supply for energy demanding units such as leaves and chloroplasts because of the maximization of the scaling of surface areas for gas exchange with the atmosphere and the minimization of the internal vascular distances for transferring available resources to different organs and tissues because of the fractal character of the trees. Now here in this table, I want to show you how different ratios between the woody surface area and the woody volume and the leaf surface area and the woody volume and the total surface area and the woody volume relate to all these tree structural metrics that we computed. So no relationship was found between these ratios and the box dimension. We do not have an exact explanation or hypothesis at the moment for why they were not related, but we also conduct some additional experiments in a different study to better understand the nature of the box dimension metric. And also we found that these three ratios were negatively correlated with all path length metrics, except from the relationship between the minimum path length and the leaf surface area over woody volume ratio, where the relationship was not significant. Now, these results indicate that as the trees increase the average distance from the ground to the leaves, as you see in the figure here, which indicates a longer hydraulic pathway, their woody volume increases faster than the surface areas of their woody organs and leaves, which actually helps us to explain the assumption of Smith et al. 2014 that the hydraulic conductance of trees reduces with mean path length. This can come from the fact of a reduced hydraulic conductivity over a longer branching network, which might come from reduced evaporative surfaces like leaf stomata and stem lenticels relative to the addition of woody volume. However, we know, according to the WB model, that in large trees, much of the volume is dead and not conductive. And based on the methods that we have used here, using the terrestrial laser scanning, we are not able to differentiate between the dead and living woody volumes because the laser pulses cannot penetrate the woody material. Therefore, it might also be possible that the leaf area and living and conductive volumes remain in balance as the tree expands its branching network. Now here I want to show you some additional results that we found because they are related to the theoretical expectations of the WBE model. So according to the WBE model, we know that trees have a fractal-like architecture, and their vascular network consists of all these pipes that are tapering from their base to their tip. And as I said earlier, there is a power function relating the woody surface area of these pipes and their volume, which is also where actually the scaling exponent is a multiple of the quarter power. Therefore, this function is also called quarter power function. In our study, we found that the branch and total woody surface areas scaled with the branch and total woody volumes with exponents 0.47 and 0.52, which were both very close to 0.5, which is the theoretical expectation for the scaling exponent based on the WBE model. Now, from evolutionary perspective, this underlines the idea of minimized outer surface area of organisms, as long as this biological pattern is consistent with the structural form of the organism, their physiological needs for gas exchange with the surrounding environment. So for the conclusions, I would like to reiterate that we used close-range remote sensing methods, terrestrial laser scanning and digital image analysis to produce detailed woody and leaf surface area data for the urban trees. This data gave us the opportunity to analyze in a more holistic way the complex dimensionality excuse me, of both the photosynthetic and non-photosynthetic components of the trees. The terrestrial laser scanning has become an important tool to quantify the three-dimensional structure of trees and their fractal architecture. Therefore, measurements of tree surface areas might be the most important new advance in tree measurements that are associated with terrestrial laser scanning, especially the surface area measurements that describe the woody skeleton of trees, because there has been less focus and studying on the woody skeleton of trees in terms of the woody surface area. So I would like to acknowledge Dr. David McFarlane, who is my academic advisor, and Dr. Dominique Seidel, who both contributed in this study. Also my doctoral committee at Michigan State University, the MSU Campus Arboretum that gave us the permission to conduct this experiment on the MSU campus, the MSU Department of Infrastructure, Planning and Facilities that assisted us during this experiment and also provided us important information for each tree that we sampled. Lorraine McIntyre, who helped with the image capture and the image analysis in the lab. Sam Clark, who is our research technician. Also the Voyukos Fellowship, which supported my time through this study. The USDA National Institute of Food and Agriculture, that supported part of Dr. David McFarlane's time. And the USDA Forest Service, FIA Northern Region, for funding the purchase of the terrestrial laser scanner. And also the German Research Foundation, DFG, for supporting Dr. Dominique Seidel's time. So here's the literature. And thank you very much for your attention. And I'm glad to receive your questions.
Okay, great. Thanks, Georgios. And just before we go on, while we're getting acknowledgements, I just want to acknowledge that Dominic Seidel is on Zoom from Germany right now. So Dominic, thanks for attending. I don't know if it's your bedtime or whatever, wherever you are right now. So yeah, Georgios will just, did you want to say something, Dominic?
Yeah, I just wanted to say thank you for inviting me, actually. And it's about to be bedtime, but I'm okay, thanks.
Okay, very good. So yeah, so go ahead and just go ahead and unmute. There's a lot of people on here. I know it's not the best way. You can unmute and ask a question if you have one right away. And you can also type questions into the chat. We'll just try to make this as orderly as possible. But Georgios, you can go ahead and field the questions.
Okay. So.
Well, that was really cool work, Georgios. Thank you very much for presenting, and congratulations on the paper. I'm really excited to read it in more detail.
Thank you.
So, it's cool for me because I think about, trees in forests most of the time, and I don't think very much about urban trees. So I'm sorry if this is a bit of a sort of basic question, but could you sort of maybe talk a little bit about, what makes, why you saw these relationships and what you might expect to be different maybe in the forest scenario. Right. One thing I did pick up on was you said, they tend to be more squat in urban environments, right. So presumably like wind speeds are higher. In some ways that sort of surprised me because I would think that maybe the urban environment would be more sheltered you know, you don't have so much sort of light competition except maybe buildings, and they sort of tend to be more cared for. So I don't know, could you sort of expand on that a bit?
This is a great question, thank you. So the urban environment is a very complex growing environment for the trees. And yeah, in a recent study, Dr. McFarlane and I have focused on the effects of the anthropogenic infrastructure on the fractal character of the trees. So we know that the urban trees, although they do not compete for light absorption with their neighboring trees because of less competition, they have to face very strong wind loads because the wind loads are amplified within an urban environment, especially for those trees that grow next to buildings, like at the edge of the buildings, where we expect the wind loads to be enhanced. And according to the based on the mechanical studies of the trees, and we know that the way that trees in urban environments cope with the strong winds is to develop wider crowns and shorter trunks, so they have this quad form to better dissipate with their branches the wind loads. Now you asked how we expect these relationships to differ with regards to the rural forest trees. So we expect that most of the woody surface area of the trees would come from the branches, because we know that urban trees have more branches and more extensive and complex branching network than rural trees that grow in a dense forest stand, for example. So this is a difference that we expected. We expect that rural forest trees, on the other hand, will accumulate less woody surface area on their branches, and we also expect that the urban trees have a more evident fractal character because of less competition by their tree neighbors. So therefore, we expected strong relationships between the woody surface areas and the box dimension, which is a metric that quantifies the fractal dimension of trees. Whereas in the rural forest areas, these relationships might be weaker. But I personally think that also in the rural forest condition, there should be correlation between their woody surface areas and their fractal dimension. But probably it's a weaker relationship.
Yeah. Oh, that's really interesting. Yeah. So maybe these are like more the ideal trees.
Right.
Yeah. Very cool.
Thank you. So we also have a question from Dr. Akihiro Koyama, who is asking, can you incorporate specific leaf area in your models? Yes, this is actually a very good question. And with the laser scanning data right now, we do not have the specific leaf area data, or with the images, with the digital image analysis, we cannot calculate the specific leaf area, but we conduct destructive sampling in the field, where we cut the trees down whenever we have this opportunity, and then we can study also the specific leaf area and incorporate this area in the models. Now, the interesting thing is that from a point cloud, from a terrestrial laser point cloud, if we want to estimate the leaf area or the specific leaf area, can be a little bit tricky, because we need to separate the leaf components from the woody components. And based on the literature and what we know from our own experience, these algorithms that exist to separate the leaves from the woody component of the trees can be biased, because they include a lot of woody material in what should be foliage. So this can be tricky as well. Okay, so then we have another question from Dr. Richard Colby, who says, do you think your results for urban trees are really encapsulating characteristics of open-grown trees? That is the important comparison between open-grown and forest-grown trees rather than urban vs rural trees? Yes, so that's a good question. So, so a tree that grows in an urban environment is not always growing totally in the open, because it can be shaded by buildings. Therefore, in our experiment, we also we also record the distance from buildings, for example. And in some other models, we consider these factors as well. But we also include in our study here, for example, we have also included trees that were growing totally in the open, in an open space, that were not close to buildings or other trees, or that were not either paved by impermeable surfaces. So, okay, yeah. So, yeah, I agree that it's not entirely the distinction between open grown and forest grown trees, but it's more accurate. It's more accurate to say that it's a comparison between urban versus rural forest trees.
We have a couple minutes for probably one or two more questions. If anyone is interested, either type it in the chat or you can unmute and.
Okay, I think Ivan probably has a question. Oh, no.
Okay, well, everybody, we can, however, virtually thank Georgios in the weird Zoom world, and for an interesting presentation, and thumbs up or wave or whatever you want to do. Thanks, Georgios, again for presenting this very detailed and interesting and really novel data. I just would make a point that this is really the first time, you know, where, I mean, within the last few years, and we're not the only lab doing this, but this is the first time that we've really had the opportunity to say that we've come close to actually measuring the surface area of a tree, the total surface area of a tree. So it's a real breakthrough. This technology has enabled us to do things we just simply couldn't do before.
Thank you very much for your attention and your feedback. That's very important. Thank you.